Hello and welcome to another episode of Talking Wild, where we look behind the scenes here at Jimmy's Farm and Wildlife Park. But today we're talking about science. We have got the wonderful Debbie and Tullis here from Nature's Safe. Welcome.
Yeah, welcome, Jimmy.
Thanks,
Now,
Jimmy.
your organisation is incredible. You hate it when I say it, but it's a bit like Jurassic Park. You get that all the time. But tell us a bit about what you do.
Okay, so we are a living biobank, which means we take tissues and reproductive samples from species and process them and store them in a way that they maintain their viability. So they are living. So the DNA is intact and the entire cell is intact, and so that we can create new life from those cells in the future, should we need to.
See, that's incredible. So you are putting life in the deep freezer.
We are.
And then you can get it out and it's still living. And that
Yep.
is tissue and cells, and particularly sperm,
Yep.
which is incredible. How did it all start? How was it created?
Well, I suppose it started a bit of passion of yours as well. It's about our rare breeds and how we could save our rare breed Suffolks. That's how it really started.
So rare breed horses,
Yeah.
it started with you.
Exactly, and it's how we could preserve our amazing heritage and cryopreserving them down in a way we can bring them back. And we thought, if we can do that to the Suffolk Punch and those other amazing breeds of horse, why can't we flick this across to rhinos, elephants?
Sure.
And, yeah.
But hang on to us. It doesn't just start like that, does it? Because you don't just go, oh, do you know what? I'm going to do this, that, and the other. When did you go, oh, I really want to freeze horse tissue? As a kid growing up, I mean, you started the whole thing. How did
crikey.
you lead into that?
Yeah, I mean, there is a backstory to it. It's quite a sad story, really.
Right.
We were naturally covering a mare one day. We were all the natural covering, and the stallion went inside her and ruptured her inside. She passed away. And I firmly believe I wouldn't be sitting here talking to you today without that one accident. We've all had things in our lives.
So a tragic accident of two horses mating, and the female died as a consequence.
And we thought we've got to do artificial insemination. I mean, it was literally as quirky as that, and we got into the AI, and we were just very lucky in the right place at the right time, and we were the first place in the UK to start doing that, and it just snowballed from that one accident. We started looking into artificial reproductive technologies, basically.
And it was so new, you had to go to New Zealand to train, didn't you?
Yes, went to New Zealand, which is where your heart is a lot of the time, isn't it?
Right, yeah. Had to bring New Zealand in there.
So we looked at, because they got the North and South Island, so the shipping horses is very expensive, so they ship semen, so we learned how to do it out there. Came back. I used to hatch chickens in incubators, cleaned all my incubators out, put all my instruments in that. So, and most probably doing everything wrong to start with, but you learn and it built up and it snowballed, and yes, here we are today.
So you started and still have a company that dealt with horse breeding, and then you thought that's worked, it's successful. Yeah. So you could take the same principle of taking semen and storing that in dry ice, and bringing it out, and then artificially inseminating. That's a tried and tested method. You know it works. So you're thinking, well, we could do this for wildlife.
Yeah.
And so from there, it's incredible. Because you have got, tell me what you've actually got. You've got how many species in the freezer?
I think it's
314, I'm getting in there before you.
Yeah, I was going to say, I already said to you ask you.
314 different species we have got
314.
safely stored.
And effectively, those animals in that tank are still alive. It's incredible. You know, they're not sort of galloping around in there, but they're still alive in that tank.
So tell me about them. What have you got?
So we've got things like southern white rhinos, Asian elephants, even coral, amazingly. You know,
that's incredible. So the sperm, and have you got eggs, or is it just tissue or living cells? What's actually stored?
We've got a mixture of tissues in there. So we've got skin cells from everything that we can get skin from, and then from some species we've got sperm and eggs and eggs as well. Yeah.
So if you've got, I get the idea. If you've got sperm and eggs, because you could take those out and you could fertilize the egg, and then you could place that egg into a surrogate, for example.
That's right.
Correct.
Yeah.
But what about the tissue? So if you've got a piece of tissue that's cut off of an animal, the animal's passed away or whatever, you cut that off. What do you do with that?
Wow.
Well, the tissue,
fascinating.
It is amazing what science and technology can do. First of all, we can re-engineer that skin sample so we can grow up and we can turn that into sperm and egg. It's already been done on mice. It's done on the human side. Have we done it in a rhino? No, not yet. But that technology is evolving. But what we do know, if we don't bank it, we lose it. So that some of the technologies have got to catch up.
Yeah.
But that's just some of the stuff. That's why tissue is so important, because it's got the whole DNA. of that animal.
And the beauty of the skin, it's like if you cut yourself and you create a scab, you know that your skin regenerates itself really quickly. And so we have grown cell lines from skin cells of white rhino the other day and the red squirrel, amongst some others. But we can replicate those cells. So you have, an infinite supply of that animal's DNA.
So once you've collected tissue from a white rhino,
Yeah.
in theory, you don't have to collect any more because you just keep growing the same cells. It's good to have a lot of different tissue for variation.
Yes.
But you can just keep growing cells.
For those, but the DNA would be the same. So we
yes.
need genetic diversity for it to be a viable population.
But you're right.
But you can, yeah. And with skin cells, every cell in the body starts as a stem cell. So that's what the tech that Tullis was talking about. Scientists can switch that skin cell back to a stem cell and then direct it to be a sperm or an egg. So then you've got reproductive samples, and you can then produce a new animal.
And would that be a clone?
No, that's not a clone. No,
that's not a clone.
That's not a clone. But what's so interesting about this is when you've got semen in straws, once you've used up your 50 straws, it's gone. Beauty about cells, you replicate, you just reproduce. You have a never-ending supply, which is just incredible when it comes to conservation. Yeah. But you do need those 50 different genetic lines. Right. So one's not good enough.
So you need a
diversity,
some good samples.
Yes.
And so where do you get all these samples from? Because it's important that you work with zoos and wildlife parks. So if an animal dies, how does it work?
So we partner with, we've got 74 UK zoos on board and aquariums and wildlife parks on board at the moment. So every time an animal passes away, we send them all the stuff that they need, all the packaging and instructions on what to take. And they take a bit of tissue for us and pop it in the post.
Literally, pop it in the post.
Well, you wouldn't want to get that delivered to the wrong address, would you? Imagine it's my Amazon delivery arrived. Oh no, it's just a load of tissue.
Testicles.
Yeah. Testicles, yes.
Well, we did, didn't we? So testicles, let's talk about that for a moment, because our lovely brown bear, Diego, he's really well represented in other collections. He's got lots of children already.
Yeah.
With the arrival of Broomhilda, obviously, she is a Eastern European brown bear. He's a Scandinavian brown bear. We don't really want them breeding, but he's getting quite amorous. He's getting quite aggressive. So just like your pet dog at home or pet cat, they go through a little castration, but we didn't want that to go to waste. So Diego was castrated, a very simple operation. Say simple, it's quite involved.
It depends who's what end you're at.
Yeah, exactly. But then I got the testicles in the car, drove them up to you.
At high speed.
At high speed.
Yes.
Safely, though. At high speed. But then we got the testicles out, we dissected them, and then we removed the semen, but some of the tissue was stored.
Yep.
And we put the semen under the microscope, and it was viable.
Yep.
And the next step, what happened to it then?
It was incredible. We froze it down to minus 196 degrees. And I don't know how you found that sort of whole experience in that sort of process right the way through. I always find it incredible. We've done this for 30 odd years and every animal's different, but when you freeze it down and you bring it back to life within 30 seconds, it's just incredible, these cryo technologies now. And obviously you were at the thick of it there, weren't you? And dissecting those testes apart to get hold of that semen. Amazing.
So getting the semen out and seeing it still viable after the car journey, I was like, amazing.
Yep.
But then putting it at minus 190, whatever it was, and then defrosting it and it's still viable.
Yeah, that's
so
right.
that means then Diego, when he passes away of old age, his potential to carry on and father new bears in the future is still there.
That's right.
But his legacy will live on in his, his semen and his tissue. It's always there for the future, which could be absolutely paramount for the survival.
It could be, and also it sort of opens the pathway really. So it's worked with that brown bear. It could work with other brown bear, other subspecies, black bears. It could work with polar bears. Yeah. So all bears. Is he the first brown bear?
He's the first brown bear. To have semen in our tank.
In our tank. How on a worldwide thing, I don't know, but certainly
with us. Yeah.
And you're right. And this can be then shared across other species. When you get a protocol, then that protocol can be pushed across. So it's not just that bear, it's other ones that he'll be saving as well.
Because that's a massive part of the work, isn't it? It's developing protocols for some of these different species, because so many have never been cryopreserved before. And so that's a massive part of our lab team's work, is working out the right protocols and the right
Yeah.
freezing mediums to use and all of that.
And tell me about the proof of concept. Have you had any major successes?
We have. So I'm gonna
Go on, Debbie.
Do you want me to
No,
talk
you
about
go for it.
coral?
Go on. Yeah, I
amazing.
know.
He doesn't. Dobson doesn't normally let me talk about coral. So just before Christmas last year, we worked with the Horniman Museum in London, and they are only one of 2 places in the UK that have spawning coral.
Right.
And they've got some of the rarest species of coral on the planet in this amazing back room at the Horniman Museum in the aquarium part. And we got the call to say, my gosh, we think they're going to spawn. So the corals spawn according to the moon cycle, but only once a year. So it was a really stormy weekend too, wasn't it? So we got the call and we put liquid nitrogen in the tank very safely and drove through the storm to London. It was all very dramatic. And set up, did some tests because it's a really particular freezing technique for coral sperm. And so we had to do some test runs the day before, and they were like, oh yeah, no, nothing's going to happen today. So went back to the hotel, and we got up the next morning, and then what's the time frame that you have it? It's full moon, 7 days.
Yeah, it's 2 hours after sunset, so they put them to bed at 9 o'clock in the morning because they're Australian coral, so of course different time
zone.
zone.
They've got them on the right time zones.
And literally, you think they're there with their watch looking at the coral art, and literally 11 o'clock, 2 hours, they started spawning.
No, I know. We were in the lab. We were just down from them. They had gone up to check. And we got this little squeal of excitement saying, yeah, they've gone. And so then it was all hands on deck. So corals spawn, so they're hermaphrodites, so they release bundles of eggs and sperm together. The eggs are really lipid heavy, very fatty, so they float to the surface. Yeah. And then when we're in the ocean, the waves will separate them and they'll go off and find people to fertilize. But with them, they were pipetting the bundles off the surface. And then they put them in a test tube. They separate out. We got the sperm out and went through the cryopreservation process.
So you froze it down?
So froze it down, yep. And 2 days later, they
thawed it out, mixed it with some eggs.
Mixed it with some eggs and got some embryos. And then we've got some coral babies.
Cryo coral babies.
Yeah.
That's incredible. So collecting the eggs and the semen, both go into liquid nitrogen?
The semen.
Just the semen.
The eggs are very difficult.
That's the next phase.
Yeah. So, but the semen froze it down.
Yep.
And then got it out, put it with the eggs,
yep.
and that created new coral.
Correct.
Yep. See, that is incredible, isn't it? Because one, it's the preservation of the coral itself, which holds its genetic diversity and we preserve it forever. But the real application of getting that semen out and then fertilizing it really shows that now you could then take those little tiny corals and you could put them back in the wild.
Yep.
Or you could re-establish a reef, whatever you want to do, is real proof of concept.
It really is, and it really provides a channel to expedite the process of regeneration as well, because you can direct that fertilization. You know, you've got the power to collect as much semen as you can, safely store it, and then, as I say, direct it so that the fertilization is more likely to happen. So you can scale up that reproductive process really much more efficiently.
So in theory, you could go around all the coral reefs that are in terrible disarray.
Yeah.
And when they are releasing their sperm and their eggs, you go and sample all of those and they can be locked away in the freezer.
Yeah.
And whenever you need them, you can bring them out.
Yeah.
And even then, maybe selecting ones that are adapted to withstand bleaching, for example.
Exactly.
And then, so you're not speed up evolution, but you're favouring those ones that would be better off surviving in the future if bleaching continues.
That's right. And opens the door for research too, that you can, if you're safeguarding some of the sperm, then you can use some of that because you've still got some left over type thing. You can use that because the Horniman are doing a lot of this work, amazing work, to see if they can create a hybrid of coral that can withstand warmer waters, for example. Yeah. So it just provides you with that opportunity that you're not having to rely on, you might lose 80% of the sperm if you did it naturally, and then you've got to wait another year to be able to try again. Whereas if you can capture it all and keep it alive, then you can do those research trials whenever you want.
And holding stuff in stasis, as opposed to having vast tanks of coral constantly breeding them, re-establishing, breeding, re-establishing. that's a big expense
Yep.
and takes a lot of time
Yep.
and man hours, where if they're in the freezer, they're in the freezer.
That's right. Doesn't take up much
But
space.
the egg's hard.
Yeah.
Why is that?
It's just that the larger the tissue is, or the eggs, the harder it is to cryopreserve. We always use the analogy: if you were to put a bottle of wine in your freezer and forgot it there, the water expands and it breaks that glass. The same happens to a cell. So if you've got an embryo, it just ruptures and breaks. So we have to find a way of dehydrating that embryo, basically. And that's what we're looking at with some really cool technology around the horizon that we're hopefully will be successful. And if that is, it just is endless then. That opens the door to so many other.
Well, I think that's fascinating, because that brings on to our collaboration, which is really exciting. So we've got a PhD student who's going to start work on it, and that's looking at the potential of taking a fertile butterfly egg and putting it in the freezer, it coming out, being defrosted, and it hatching into a caterpillar.
Yeah.
Now, I know there's been some work done on silkworms, silk moths, so in the silk industry, very valuable. But there's been some success, so there is potential at working. But if that does work, then it's a huge win for conservation.
Oh, it will
That's
be
massive.
stratospheric. I can't tell you, obviously, Jimmy, if we could really sort that out, because that side, or sort of the insect side, is really difficult to do. Some of these these eggs and the larvae in that side. And my God, this will be revolutionary if we can.
But they're vital, aren't they? I mean, to our health of
Absolutely. And I think the other applications is that not only could you breed lots and lots of rare butterflies and store their eggs ready to hatch whenever you wanted them, but you could then apply that to farming.
Exactly.
And you could breed lots and lots of predatory insects. Their eggs could be stored.
Exactly.
And you could spray them onto a field.
That's right.
Instead of pesticides.
That's right.
You put insect eggs out.
Exactly.
Oh, we're really thinking outside the box now.
Well,
I think it's
And
great.
if you don't like creepy crawlies, it's going to be terrible. But it's fascinating. But I've got to say that this is not cheap technology. It's a huge expense, not in terms of the man hours you put in, but also the equipment that you need and the space you need. Funding. So what things have you done to try and fund it? Because you're a charity, aren't you?
We're a charity. We rely on the goodwill of people for donations 100%. It's the people out there, the businesses, the trusts, the grants, and all that really keep us going. And so we can't thank people enough, because it's imperative. We're always fundraising. The more we fundraise, the more work we can do, the more research work.
What things have you done?
So we climbed Kilimanjaro last year.
Right.
There's 4 of us went up there, and one of the ladies who's an ambassador of the charity, she was blind.
Yeah.
And we raised £50,000 for the charity
Wow.
doing it, and it was incredible. And one of our trustees came up with this, Tony, as well. So it was amazing. And I had the fortunate thing afterwards to go to see the last 2 Northern Whites straight
Did you?
afterwards, and that was the most emotional. I'm emotional best of times, but it was just incredible.
So tell me about the last 2 northern white rhinos on the planet.
Yeah, mother and daughter.
And what's the hope for them?
They've got about 30 embryos banked already, and they're hopeful to put those into southern white rhinos as like a surrogates.
Yeah.
But one of the one female is totally infertile. The other daughter, because it's mother and daughter, you see, and I think Sudan died in 2017, the last male, but they've got semen banked from him. So it's difficult because there's only 2
Isn't
left.
it terrible? There's 2 left, and one's not fertile.
No.
I think they're both infertile, actually. But yeah.
And you just see those last 2 animals, and then without this technology,
that's that.
That's that.
Yeah.
It is. And
Forever.
And when you were there, they don't know they're the last 2. And it was just incredible. See, we as a species have done this. And we shouldn't, there's something that should never have happened.
No. All those millions of years of evolution.
That's right.
Perfectly adapted to their environment,
Yeah.
and then reduced to just 2.
Yeah.
In such a short space of time, when you think about it, really, as you say, millions of years of evolution wiped out, really quickly.
In a lifetime.
Yeah.
In our lifetime.
Yeah.
And the Science magazine, brought a great thing that last year, had a picture of them and saying skin cells could be a way that we can actually help save these species. That just shows the proof of concept what we're doing. And that, yes, it's going to be hard for those 2, but we've got to make sure we preserve all the others.
Well, learn from this, isn't it?
Yeah.
And try and make sure that doesn't happen again. So
from what you're doing, if those skin cells are preserved, in theory, as the technology catches up, you would be able to produce sperm and egg
Yeah.
and produce an embryo that could be then put into maybe captive rhinos
Yeah.
here.
That's right.
Exactly that.
Yeah.
And you've got to think of all your trees and plants, the Millennium Seed Banks, all that's all stored. But where's the one for animals?
Yeah.
Or species. So you're right. This technology could be absolutely critical for many of our species out there.
I think it's fascinating, and long may you continue, because we need you. We need you to carry on doing your work. And if you want to find out more, you've got to check out Nature's Safe, get on their website. There's loads of ways of donating and finding out more information on what these guys do. It's truly incredible. But I will see all you guys again for another episode of Talking Wild.